Hello friends, this is Sonu and today we'll discuss about the microbiology. In last time as I discussed that was the introduction of the microbiology, types of the microorganism and some discuss about the prokaryotic and eukaryotic differences. So today we'll continue about the bacteria. First we will discuss about the bacteria, then we'll move further different microorganism. So first of all, what is the bacteria? Bacteria, it is a large domain of the prokaryotic microorganism because all bacteria are prokaryotic. So that's why it's a large domain of the prokaryotic. So you can see this is the very simple structure of the bacteria. That you can see that this is the cell membrane or plasma membrane. That plasma membrane also in our human body that made by the lipid bilayer. So the same plasma membrane. Inside the plasma membrane, this is the DNA material, or we can say chromosome. This is the plasmid. This is the cytoplasm, and the cytoplasm water and enzyme and so on are situated there. Then this area, this is the, we can say, flagella, and this black and blue gapping, that is the cell wall. This is the plasma membrane, as I said, and this is the hair-like structure, that is the pili or fimbriae. And then the red and blue gapping area, that's a capsule, or we can say also at the slime layer. So first of all, this is the basic structure of the bacteria. We can say that very basic structure. First of all, what is the flagella? Flagella, the function of the flagella. As we know that flagella is the we can say it's a maintain of the motility of the bacteria, or we can say movement of the bacteria. Means by the flagella, bacteria can be moved. This flagella, and draw the simple structure. This flagella can be this side, can be this side, or can be all the bacteria. It depends upon the types of the bacteria. And one function also, that it's a sense the chemical and temperature in the outside of the area. So movement as well as sensation. Second one, about the pili and fimbriae. Actually, it is the hair-like structure, and the function of the pili and fimbriae, we can say that it's a help to adhesion, or we can say it's attach the mucous membrane, as well as it's the information, or we can say transmit of the genetic material, genetic information from one bacteria to other bacteria. That function of the pili and fimbriae. And the capsule or slime layer. Capsule, first of all, it's a we can say polypeptide, that it's we can say help to maintain or help to attach our mucous membrane of the human body, as well as it also prevent by the we can say protect by the phagocytes, or very simply you can say that skip the phagocyte, because phagocyte can't be engulf that bacteria who have capsule. So by this way you can understand that it's a very pathogenic. So flagella, pili and fimbriae, and capsule, or we can say slime layer, these three component, it's a very pathogenic component. By this component, bacteria can be very pathogenic, or it can be damaged too much of the human body. Then plasma membrane, you can say that this is the plasma membrane, and the cell wall. So now we will discuss about very deeply about the cell structure, because cell structure very important, because of the lot of antimicrobial drug are act on the cell, or destroy or inhibit the cell structure formation. That's why we should know that about the cell structure. So let's discuss about the cell structure. About the morphology, we already discussed in a last time, but very briefly we will try to discuss. Morphology, or we can say shape. Three main shape we have. First, cocci, then bacilli, or sometimes rods, and then spirochete. A spirochete. These three are the main morphological structure. But in some bacteria have different types of tendency of the we can say shape, that can be comma shape or curved shape. that is the cholera. Cholera bacteria has comma shape or curve shape. So cocci, it can be diplococci if double structure, if long structure, that is the streptococci, if the cluster formation, that is the staphylococci. The same thing in a bacilli and spirochete. So this is the very basic about the morphology. Whenever we will discuss the individual bacteria, we will discuss very deeply about the shape and size. So now you just remember that most probably bacillus, cocci and spirochete, these three shapes are mainly found about bacteria. Now we will discuss about the bacteria. First of all, bacteria is divided into two huge groups, that is the gram positive bacteria as well as gram negative bacteria. So why the gram positive bacteria is called gram positive, why the gram negative bacteria called gram negative? Let's discuss. So first we will discuss about the bacteria cell wall. For example, this is the bacteria, this is the bacteria cytoplasm or cell wall, plasma membrane, and outside of the bacteria this is the layer. And this layer attached by each other. Like that, attached. It's not very good diagram, but you can understand. So by this attached, a lot of is there, just for understanding purpose, you can understand. So this is the cytoplasm, this is the plasma membrane, and outside of the plasma membrane, this is the layer, and this layer's name is peptidoglycan. Peptidoglycan means this layer is made by peptide and glycan. What is the peptide and glycan? We will discuss. So what is the first of all function of the peptidoglycan? This is the hyperosmotic. This environment, this we can say cytoplasm, plasma membrane, is the within the bacteria physiology. I mean, if you talk about the physiology of the bacteria, then it's a hyperosmotic, or we can say hydrophilic. Hydro means water, philic means love to bind the water. For example, and the outside area is the water is very high amount. For example, if bacteria in the stomach, if bacteria in the, we can say in the saliva. So in a saliva, a lot of fluids there, a lot of water is there. So for example, if bacteria in a saliva, so surrounding area, that is the external area, a lot of water is there. Means that water will have to come or should to come in this environment, inside the bacteria, from intra to extra. Why? Because it's a hydrophilic and hyperosmolality, and inside the outside of the area, that is the hyperosmolality. That's why water loves to go inside the bacteria, and whenever water will go inside the bacteria, bacteria becomes swell and it becomes burst. So if water will come inside the bacteria, so bacteria will swell because water is too much accumulating, it becomes swell and burst, or we can say rupture, and bacteria will die. So the peptidoglycan, we can say it's a prevent by the burst, so it's prevent to the burst or rupture. So the main function of the peptidoglycan, that it's a burst and prevent or help to by the burst and rupture. So this function of the peptidoglycan. Now we'll discuss about that how it's make, which types of molecule are here to make the peptidoglycan. Then we'll discuss about the main differences between gram negative and gram positive. So let's start peptidoglycan. Peptidoglycan. It's divided into two parts: peptide and glycan. So let's peptide is a peptide. You can show it's part of the amino acid. Let's discuss about the glycan. Glycan. What is the glycan? Glycan is a unit of the sugar. Unit of the sugar. And this unit, for example, you can say this two monosaccharide. So this is two monosaccharide, and this two monosaccharide has two important molecule. First, NAG and NAM. What is the NAM and NAG? NAG is the N-acetyl muramic acid. N-acetyl muramic acid. And the NAG is the N-acetyl glucosamine. So N-acetyl muramic acid and N-acetyl glucosamine. This two component is made by the glycan as a peptide. Then it's finally made by the peptidoglycan. Now we'll discuss that how it's made, how it's synthesized inside the cytoplasm. For example, like the baby bacteria. When bacteria it's very recently it's by the binary fusion it's a replicate and make new small we can say baby of the bacteria. So inside the baby of the bacteria, how peptidoglycan is synthesized, and then how it's finally make in the, or we can say inside the bacteria, I mean external layer of the bacteria, how peptidoglycan will be appear. So for example, this is bacteria. This is a baby bacteria. In a baby bacteria have very low, or we can say very thin layer of the peptidoglycan. So just make very thin layer of the peptidoglycan, or we can say one layer. As you can say that it's a attached. This is the peptide link which attach. So here is the NAM and here is the NAG. One basic information that you have to know, that is during the polymerization, or we can say polymer which made by the cell, during the made of the any cell by the polymer, or we can say during the polymerization, we need precursor molecule to as a carrier substance, as a carrier component. So here is the carrier component, that is the UDP, uridine diphosphate. This two is the carrier protein to make the cell, because it's a peptide polymer. And here is the amino acid. This is the amino acid. And the last two amino acid, this is the D-alanine. And these two amino acid, and here is the one enzyme. This enzyme, first of all, inside the cell, it's a cross links each other, NAM and NAG unit. And whenever it's crossed or will attach NAM and nag, and then it will shift outside of the cell or outside of the cell, and then it's further moving and further making the peptidoglycan. This enzyme or protein name called bacterioperinal phosphate. Bacterioperinal phosphate, or we can say BPP, the shortcut name. Again, I will repeat that NAM and NAG unit. This is the, I'm talking about when the bacteria is the just replicate, and baby of the bacteria will try to make the big peptidoglycan for the prevent by on bursting or on we can say rupture. Due to the rupture, it's start to make new peptidoglycan, or we can say the thick layer of the peptidoglycan, because baby bacteria have very thin layer. So by the name NAG unit, it's a chain of the amino acid. I'm talking about that how it will be the shift from the inside to the external layer. The last part of the this amino acid, whenever it's attached to the layer of the, we can say plasmamembrane, in the plasmamembrane has special protein or special enzyme, that name is the bacterioperinal phosphate. This bacterioperinal phosphate first inside the bacteria, it's make, or we can say attached to each other, NAM and NAG unit, and whenever it's attached already, then it will shift in the external layer, outer side of the layer of the bacteria. This is the how the NAM NAG unit synthesize, and how it's we can say shift outside of the or external layer of the bacteria. Now we'll discuss about the NAM and NAG unit in a very we can say big diagram. Then we'll discuss gram negative bacteria and gram positive bacteria. NAM, this is the NAG, then here NAM, NAG. You can say this is the peptidoglycan layer, and by each peptidoglycan layer, it's attached by the peptide link. This peptide link is transpeptidase by the enzyme of the transpeptidase. This enzyme to help to the link each other. So one enzyme that found in a saliva, that is the lysozyme. So because in our saliva a lot of bacteria is there, but it's not harmful. Why? Because our lysozyme enzyme is protect for those bacteria who is living in the lysozyme or in the mouth. So by the lysozyme can be destroy this enzyme, destroy by lysozyme. So if the lysozyme will destroy this enzyme, means this enzyme will not link peptidoglycan each other, and peptidoglycan will not live like a unity. So it will be individually, or it will be weak. And as we know that if peptidoglycan will not strongly bound or strongly protect in outer side of the bacteria, then water will come, bacteria will swell, and bacteria will rupture, and then bacteria will finally die. So that's why any drug, for example, the drug is penicillin. So penicillin can be inhibit this enzyme, or it can be inhibit to the attachment of the peptidoglycan layer, or by the lysozyme can be destroy this enzyme. So you can understand, or you can remember that it's very important, we can say the attachment. By this attachment, it's attached by each other, and it's very tightly bound and tightly protect the bacteria. Otherwise bacteria will, by the osmotic burst, bacteria can be die if this peptidoglycan will not tightly bond each other and not will protect. So this about the bacteria, and I mean layer of the bacteria. Now we'll discuss about the gram positive and gram negative. That why named gram positive and gram negative. Gram positive and gram negative. First this is the cytoplasm. This is the cytoplasm of the gram negative bacteria. Here, as you know, this is the peptidoglycan each other. You can understand that it can attach to each other by the peptide linkage. In the gram negative also the peptidoglycan layer, but a very thin amount, very thin, just 1 to 2 layer. But in a gram negative bacteria has additional feature, that is the additional cell wall. This is the additional cell wall. But in a gram positive don't have additional cell wall, just one cell wall, that is the cytoplasm, which present in every bacteria. So in this bacteria also we can say plasma wall, plasma membrane. So in a gram positive bacteria just one cytoplasm, and in after the cytoplasm that is a thick peptidoglycan layer, but no any additional plasma membrane. This is then a gram positive. But in a gram negative bacteria has a special protection, that is the additional cell wall, which made by this cell wall is made by lipid and polysaccharide. So name is the lipopolysaccharide. Lipopolysaccharide. It's made by lipid A and polysaccharide, means many saccharides. So lipid A and polysaccharide. That's why the name is gram negative and gram positive bacteria. That is the main differences. We can say the huge differences that you can identify that this is a gram positive and this is the gram negative, because in every gram negative bacteria must we have the outer membrane, that is the which made by the lipopolysaccharide. And lipopolysaccharide is the lipid A and polysaccharide. Lipid A, that is the very toxic, or we can say that is the main virulence factor of the gram negative bacteria. Lipid A. That's why you can listen or you can see that in an intensive care unit a lot of people die due to the lipid A, because very toxic substance, very lethal. Patient can die very, you can say early if treatment will not start properly due to the lipid A, very toxic molecule. So that by this way the gram negative bacteria is very, you can say very potent and very intensive protect by this layer. But in a gram negative bacteria is not protected well instead of the gram negative bacteria, because of it don't have the additional cell wall, just thick peptidoglycan layer. So lipopolysaccharide also, or we can say endotoxin means whenever you listen that endotoxin, 100% absolutely you suspect that it's a gram negative bacteria, because the lipopolysaccharide, that's the other name or synonym name, that is endotoxin. But in a gram negative bacteria this is the exotoxin. But exotoxin may produce gram negative bacteria as well as may produce in a gram positive bacteria. That's why we can't say that if you listen or if you see that this is the exotoxin, that you can suspect that it can be gram negative, it can be gram positive. But if you see the endotoxin, means absolutely it's a gram negative bacteria. That is very important to know. Now we'll discuss about that main differences, like what is the main clinical role or other important differences about the gram positive and gram negative bacteria. positive and negative. Also endotoxin. Endotoxin. So let's first gram positive bacteria that absent of additional cell wall. Absence of additional cell wall. Then here extra cell wall. Extra cell wall. Next difference. Yeah, one thing I forget to mention that in the gram positive has teichoic acid, like in a gram negative has lipopolysaccharide. The same thing, like the gram positive have teichoic acid. So this is the cell wall, I mean the plasma membrane. This is the peptidoglycan. In the peptidoglycan, this is the teichoic acid. Teichoic acid, or we can say lipoteichoic acid. Lipoteichoic acid. Actually, the teichoic acid and lipoteichoic acid are still unknown that what is the main function of the teichoic acid and the lipoteichoic acid. But in some book, in some author said that it's also help to the during the replication, during the we can say the binary fusion, and sometime also it's help in during the bacteriophage. Means whenever the virus it's on GNM, the virus GNM material it's inject inside the bacteria, that is called bacteriophage. So on that time it also we can say help. This is the function of the teichoic acid, but still exact unknown. So one difference also we can see that the gram positive has teichoic acid. Teichoic acid. And the gram negative has lipopolysaccharide. Another it's a heat liable. Heat liable means in a 60 degrees Celsius bacteria can be dry, because it don't have any extra layer, so it's just made by the protein, we can say polypeptide. It is the protein, the layer, this is the part of the protein, and protein can be denatured by the heat. That's why the heat liable within the 60 degrees Celsius bacteria can be dry. And this is the heat stable. Heat stable means in 100 degrees Celsius, in a 1 hour can be it's a pathogenic, it can be survive. So the also the very main differences. Then it's a less lipid compound, because of it don't have any extra cell wall just except plasma membrane, but it's a very rich lipid molecule or lipid compound. Then in gram positive bacteria, or those bacteria who release the exotoxin, all gram positive bacteria are released or synthesize exotoxin except one bacteria, that is the Listeria species. So Listeria species is gram positive bacteria, but it synthesizes endotoxin. That is the exception. But all gram positive bacteria are synthesized or release exotoxin. So we can make vaccine for the gram negative bacteria. For example, tetani, Clostridium tetani, that is the gram negative bacteria. And as we know that we have anti-tetanus vaccine. Coronabacterium diphtheria, BCG, we have vaccine because it's all gram negative positive bacteria and it's release exotoxin. That's why we can make the vaccine. So we can make vaccine, but in a gram negative case we can't. We can't due to the lipid A and extracellular wall. We can't make vaccine. We can't make vaccine. So this is the main we can say main differences. Some other differences also that the gram negative bacteria have a special porine molecule, porine channel, but in gram bacteria don't have porine channel, because due to the porine channel, what uh, because the gram negative bacteria, inside the bacteria which produce the endotoxin, it can be decided that which types of molecule should be go outside or which types of molecules should be come inside. Why? Because the bacteria have an additional layer, that is a lipid bilayer, and that lipid bilayer can be decided that it should be go or it should be not go. But in a gram positive bacteria don't have any extra cell wall, so we can say don't have any porine, just make layer the peptidoglycan. So maybe extracellular molecule can be very easy to penetrate inside the cell because you don't have extra any cell wall, and some other differences. So this is the main differences we can say. So as well as gram positive and gram negative bacteria, as well as ectotoxin and endotoxin. So we should write endotoxin. So you can make difference here as well as gram positive and gram negative with ectotoxin and endotoxin. The both can be, in both conditions these are the main differences. Ectotoxin. But just one thing you have to remember, that ectotoxin can be synthesized or can be released as well as gram positive or gram negative. But endotoxin always, if the endotoxin is there, it means absolutely it's a gram negative bacteria, no any gram positive bacteria. But in exotoxin case, it can be gram positive or it can be gram negative. So it's depend upon the laboratory investigation, that by the laboratory investigation or by the staining method, we can say that no, it's a gram positive bacteria, but which bacteria, particular we have to identify. But if the endotoxin, if the doctor or you will suspect that it's an endotoxin, means always gram negative bacteria. So these are the main differences, or main we can say about the bacteria morphological structure, that very important to know. So by the morphology we can understand that this is the gram positive and this is the gram negative. Some additional information that we should discuss, that is the name bacteriophage. First of all, what is the bacteriophage? Bacteriophage. For example, this is the bacteria, and bacteriophage, for example, this is the bacteria, this is the glycan layer, and if the gram negative bacteria, so additional glycan layer. Bacteriophage means this is the circular genetic material, this is the plasmid. Bacteriophage means some viruses is attached to the bacteria. For example, this is the viruses. This is the viruses. So some viruses can be attached to the bacteria, and it secrete the DNA material, gene inside the bacteria. And this DNA or this gene will bind to the we can say the bacteria DNA and will replicate and synthesize exotoxin with the virus's DNA. So bacteriophage means we can say that bacteria also suffering from, or we can say just like the human suffering from the bacteria. The same thing, bacteria also can be suffer from the viruses injecting the DNA material or gene inside the bacteria, and by the bacteria on replication, it's mixed with this gene or this gene, and then it's produced a toxin, that is the exotoxin. One thing also that for example, that some bacteria, like this is two bacteria, the name is the same bacteria. This is a Corynebacterium diphtheria, and this is also Corynebacterium diphtheria. But maybe this bacteria will produce exotoxin, but this bacteria will not produce exotoxin. Why? What's the reason? Maybe these bacteria don't have any bacteriophage, means this bacteria will not attack by the viruses, but this bacteria will attack by the viruses. That's why it's produced toxin, exotoxin, and this exotoxin is more lethal, more dangerous than this one. So this is the about bacteriophage information. But whenever we will discuss about the particular any bacteria, on that bacteria will suffering also by the bacteriophage. On the time very deeply we will discuss everything about the bacteriophage. Now just very basic thing that about the bacteriophage you have to know. And one thing also I have to mention about the exotoxin, approximately 100 times of gram negative bacteria can be lethal, but within 1% of the exotoxin can be very dangerous. Why? What's the reason? Let me discuss. Exotoxin is a very danger as compared to endotoxin. Endotoxin, even 100 bacteria can be start very lethal or very dangerous, but even one bacteria or 1% of the exotoxin is very harmful. In a patient can be die. Why? For example, this is our cell, any particular cell. In this cell, any bacteria, any gram positive bacteria is attacked, and there is exotoxin. But this exotoxin, maybe bacteria is die. For example, bacteria is die, or bacteria will destroy by own immune system, or it may be by the drug it will die. But exotoxin it already releases. This is the exotoxin. And this exotoxin can be moved in all over the body. It can be spread all over the body by the blood or by the lymphatic system, and maybe it can be go, for example, in a heart. So, for example, Corynebacterium diphtheria or beta hemolyticus streptococcus. This bacteria, it releases exotoxin, and this bacteria can be go in heart, and heart maybe heart cardiac problem, or maybe sometimes cardiac failure. Why? In heart you can't any find bacteria in the heart area, where, or for example in any organ, that organ is affected by the exotoxin. You can't find any bacteria or any gram positive bacteria or any gram negative bacteria that are situated or are found in that area where the inflammation or pathology is going on. But you can find exotoxin. It means what? So exotoxin can move all over the body. Maybe bacteria will not move, but exotoxin is moved. That's why exotoxin is very, very, we can say very dangerous. Even 1% of the exotoxin, that is the Clostridium tetani, it can be going in spinal cord and interruption of the or disruption of the spinal cord function. So patient can be paralyzed, or patient can be very jerk moving during the tetanus. That's why exotoxin is very, very lethal condition as compared to endotoxin. So I think this is the about all information about the morphology and the cell structure. That is very important to discuss. In the next time, in the next session, we will discuss about the staining method. That during the laboratory investigation, how we will identify that this bacteria belong to the gram negative and this bacteria belong to the gram positive. Means what is the color, and but what molecule we should start, and how we will check in the the normal microscope. So you will understand and you will like. In next session we will try to very soon discuss. So thank you so much.